Hey guys. So at this point you should have watched the video at this link that goes through the making of the fittest and natural selection in humans. In particular, this clip looks at sickle cell trait and sickle cell disease and the discovery of the inheritance patterns. And so what I'm going to do is kind of go through the practice problems on this worksheet with you. This worksheet's linked in Blend. You can print it out, or you can just modify it digitally, or you can do it on a separate sheet of paper. I'm going to go through these. I would encourage you to pause and then try to answer all of these questions on your own, and then you can hit play and I'll go through the explanation of how to work the problem so you can check yourself. That would be my suggestion for the best way to kind of proceed here. So if you read through the background information, you'll see that there's kind of a codominance going on here. You have this sickle cell anemia. You can either have, they're calling it AA for normal hemoglobin, and then we're going to use the letter S for the sickle hemoglobin. So if you're heterozygous, you're going to be AS, and we say that you have sickle cell trait, but not sickle cell disease. It's a less deadly combination, I guess. You have some cells that will remain normal while others will become sickled. And then if you have both of the sickled alleles, then you have sickle cell disease. There's something called the heterozygous advantage in certain environments, because while having any blood cells that will become sickled in a low oxygen environment would be bad, it actually provides you some immunity to malaria. So being heterozygous, if malaria is present, actually becomes advantageous. So here I'm just going to fill in my genotypes to go with these phenotypes. So we're going to say AA if you're just normal in high and low oxygen environments. I'm going to do AS. You can use any abbreviation you want, but this is what I'm going to use, the notation. And then for sickle cell disease, that would be somebody who is homozygous for the sickle cell trait. So we're just going to go through these. So this first one, if two people with sickle cell trait have children, so sickle cell trait, that means that they're going to be heterozygous. What is the chance that this child will have normal red blood cells and sickle cell disease? So basically you have two parents who are both heterozygous. So I'm going to fill in my Punnett square. They could either contribute the normal allele or the sickle cell allele, and it will be the same for both parents. And then here, I'm just going to fill that in. And so the probability of having normal red blood cells in high and low oxygen environments is going to be 1 out of 4, or 25%. The probability of having sickle cell disease in this case would be the same, 1 out of 4, or 25%. Now if they asked about sickle cell trait, then that would be 50%. Which is what they do right here. So that's going to be 50%. So now here you're getting some questions like, what are the chances that these parents will have 3 children who are homozygous for normal red blood cells? Okay, so there's a few things going on there. Each child is going to have a 25% chance, or 1 out of 4, probability of being heterozygous, right? So we're talking about this right here. And so every time they have a child, it's always going to have that 1 out of 4, 1 out of 4, 1 out of 4. So here, if I multiply these, so 1/4 times 1/4 times 1/4 is 1/64, 1 out of 64. Or if you do it in decimals, it would be 0.0156 or 1.56%. Remember, if they're asking you about the probability of having 3 children in a row, you're going to use the multiplication rule. So this times this times this. So here, we're going to kind of do the same thing. If they're asking what are the chances that these parents will have 3 children who have sickle cell trait. So you have 1 out of 2 in this case, or 50%. And you're going to have that for each child. So 1/2 times 1/2 times 1/2 is going to give you a 1 out of 8% chance that they will have 3 children with that characteristic. And this is kind of a good way to go about solving those problems. So if they're asking what are the probabilities that they would have 1 child, every time they have a child, it's the same probability, but asking if for all 3 of those things to occur, then you would go with the 1 out of 8. All right, so let's move on to the next one. An individual who has sickle cell trait has children with an individual who does not have the allele. So somebody with the trait, so that's somebody who's heterozygous, crossed with somebody who does not have the allele at all, so that's going to be homozygous for the normal. So now we're going to show the Punnett square. Again, this parent can either give an A or an S. This parent is only going to be able to contribute those A's. So A, S. Yeah. So what are the chances that any of this one couple's children will have sickle cell disease? Well, they're pretty fortunate that since only one parent has the allele, there's no way for them to have a child with 2 copies of that sickle cell allele. And so I'm going to just say there's a 0% chance there. If this couple lives in the lowlands of East Africa, what are the chances that one of their children would be resistant to malaria? So the children that are heterozygous, so these 2, would carry the sickle cell trait and be resistant to malaria, where these 2 would be susceptible. So in that particular environment, this is actually going to be advantageous. So that is 50% or 1 out of 2. All right, so remember, you probably want to pause me, go to the next question, try to answer it, and then come back and see if you got it right. So here we've got sickle cell disease, a woman with sickle cell disease. So this individual is going to be 2 S's, has children with a man who has sickle cell trait, so that's AS. So genotypes of the parent, mom is SS, dad is a carrier. What is the genetic makeup of the gametes that the mother can produce? So this is mom. She can only produce egg cells with an S for the sickle cell gene. The gametes that dad, well, he can produce them with A, or he can produce sperm cells that have S. And these are really what you're putting in those Punnett squares. They're the possibilities from those parents. So let's see, we're going to make ourselves a Punnett square here. So mom can only contribute that S. Dad can contribute a normal allele or a sickle cell allele. And so we're going to have 50% or 1 out of 2 that are going to be AS and 50% SS. So the chance that they'll have the sickle cell disease would be 50%. Which of their children would be more likely to survive in East Africa where malaria is present? Those would be these individuals here, those heterozygous individuals, because they would be resistant to malaria, which is prevalent. Okay, so hopefully you're feeling pretty good about those crosses and you feel pretty comfortable. Now we're going to move to something a little bit more complicated. Not crazy, just a little bit more complicated. So here, and I'm just going to grab a different piece of paper so that I can write big. In humans, blood type is a result of multiple alleles, so you have the A, the B, and this little I is going to be like an O, right? And it's an example of codominance and multiple alleles. So the A and the B are going to be codominant, whereas A and B are both dominant over O. So here, let's take a look at this one. So we've got two parents who are heterozygous for type A blood. And I probably do have space to write this over here. So if they're heterozygous for type A, that means that mom and dad are both IA, little I, and they have sickle cell trait. So I'm going to go back to that notation right there. And so I've got my AS. So now I want to look at the possible gametes. So that's where you're going to FOIL it, right? So that would be first, outer, inner, last. And just take your time. So I'm going to just do this right here. So first, they could have IA, A, outer IA, S, inner, OA, and O, sickle. Try not to get confused with the A's in this case, knowing that they represent they're related to the sickle cell trait, not blood type. So now these are the possible gametes, and since both parents have the same genotype, both of them are going to produce these 4 possibilities. So I'm going to fill that in on my Punnett square. And then I am going to slowly go in and fill in the possible combinations. Okay, let's see. So my possible combinations would be, let's see, type A and normal, right? Type O and normal. Type A and heterozygous, type O and heterozygous, type A and sickle cell disease, and type O and sickle cell disease. And we're actually going to use this information in a problem later, so it is totally worth it to go through all of these steps and figure out how many of each we're going to have, or what the probability of each is. So I'm going to go in and find all of my type A and homozygous. So let's see, I've got A, So much better when you guys are here to point the ones that I miss out to me. All right, so I think I got all of those. So I've got 3 out of 16 for that. Let's see. Type O and AA. So let me just find my, here's 1. And that's it. So I've got 1 out of 16 there. Let's see what's next. Type A and heterozygous for sickle cell trait. So there's 1, 2, 3, 4, 5, 6. So I've got 6 out of 16 here. Let's see, type O and heterozygous. I think those are the only 2. So 2 out of 16 here. Type A and sickle trait. Let's try something. So here's 1, 2, 3 out of 16, and then type O and the sickle cell trait. I think there's just the 1. Yeah, 1 out of 16. So those are your percentages for that one. There's actually a different way to solve those kind of problems, and it's really up to you which one you're more comfortable with, but I figured this would be a good chance to kind of go over how you might do that if you come across these. So, let's say that in this case we have a mother who is heterozygous for type B blood and has sickle cell trait, and the father is type AB and also has sickle cell trait. So in this case, instead of combining everything, we're going to look at the probabilities for the individual things. So in this case, blood type separately from sickle cell trait, and then we can use the multiplication and addition rules to figure out the probabilities for particular combinations. Similar to what we did with the children earlier, having 3 children with a particular trait. So let's see. Mom's heterozygous for type B and dad is type AB. So dad is going to be IA, IB, and mom is going to be type BO. And then both are heterozygous for the trait. So AS, AS. So I'm going to solve these separately. Alright, so in this case right here, I have 25% chance of having a child that's going to be AB. So 1 out of 4 for AB. 2 out of 4 for B. And 1 out of 4, I can't write today, sorry. 1 out of 4 for type A. Here, is that right? And then here I've got 1 out of 4, 2 out of 4, 1 out of 4. So let's look at the question. So what are the chances of a child would have type B blood and sickle cell trait? So if I look, there's a 2 out of 4, or 1/2 chance that they're going to have type B blood. And sickle cell trait, well, those are these 2 right here. So again, that's 1 out of 2 or 2 out of 4. And since they asked that and, we're going to multiply those. So 1/2 times 1/2 is 1/4, or a 25% chance. What are the chances that the child will have type AB blood and not have sickle cell disease? So I'm going to interpret this type AB blood. So there's 1 out of 4 chance there. And not have sickle cell disease. Well, this child has sickle cell disease. They didn't say anything about not carrying the sickle cell trait, and so I'm going to say that there's a 3 out of 4 chance. or 75% chance that they won't have the disease. They may be a carrier, but they won't have the disease. And in this case, if you work the math, that's going to be 3/16. Here, what are the chances that the child will have type B blood and sickle cell disease? Again, we're dealing with these n's, right? So you're multiplying. If it said or, then you would add the probabilities. So in this case, type B blood, if I come over here, that's 1/2 times 1/4, and that gives me 1/8. You could also do that in decimals, so really it's up to you, whichever you're more comfortable with. All right, so again, you should probably pause me here, go read through your pedigree problems, see how far you can get, and then once you've answered them, come on back and we can go over it together. All right, so in this one, we're using a pedigree. So remember, these are kind of like genetic family trees to follow the sickle cell disease through a family. Here, if an individual has the disease, we're not saying trait, we're just saying they have the disease, so full-on sickle cell disease, that's going to be SS. However, if they do not have the disease, that could be somebody who's homozygous or heterozygous. So what I always do is start with what I know, right? And remember, the squares are male, circles are female. So here, I know all of these shaded in individuals are going to be SS. Now, if they're not shaded, they could be either homozygous or heterozygous. But all these couples here that have children, well, if this child has 2 S's, if dad was homozygous for that allele and mom was homozygous for this allele, there would be no way for them to have a child that actually showed that SS. So the only possibility is that dad is heterozygous. Sometimes you have to do a little digging, and sometimes you just don't know based on what you're seeing. So here, they're either going to be carriers or they're going to have the disease. So dad must have been heterozygous, and because mom can only contribute that S, then I know both of the children have at least one S, and they're normal, then they're AS. And again, you have that same thing going on here for that square. So based on that, you should be able to answer all of these questions. For this one right here, for D, if the couple in the second generation has another child, so we're talking about this couple right here, remember that they're always going to have the same probability, so it's always going to be that 50% chance. And then you would have 50%, and this would be 0 because they could not have that child. All right, so following that same kind of idea here, I know this individual, I know this individual, and I know this individual. And if I have children that have the trait, then I also know these, that the parents both were carriers or were heterozygous. Who else? I know that all of these children are heterozygous because mom, again, can only contribute that S. But the rest of these individuals, I don't know. I am not sure, right? So this individual could be homozygous, or they could be heterozygous. And that's because none of the children in this generation are actually showing the disease. So I know they're all carriers because of mom, and I know that they got a big A from dad, but unless I have more information, I really couldn't answer it. So sometimes you just have to accept that you don't know. And so you don't always know all the answers, but you solve as many as you can. I'm going to go and do this one with you too, this pedigree. So up here we were interpreting them, and here we are going to draw one. These are kind of fun too. So imagine you're a genetic counselor and a couple is planning to start a family, and they come to you for information. So Jerome was married before, and they have a daughter with sickle cell disease. We're going to assume that he does not have the disease. So we've got Jerome, who would be square, and his first wife have a child, so they have a daughter who has the disease. Jerome and his wife must be heterozygous. They get divorced. I'm going to just do that. He is remarried, and they want to know what are the probability of their children. Okay. The brother of his current wife, Michaela, so this is Michaela. So she has a brother. Died of complications from sickle cell disease, so he definitely had it. But both of their parents her parents did not have the disease, but because her brother had it, they must have been carriers. So we know Jerome, and we know her brother was SS. And we know the parents must have been carriers because they don't have the disease, but they have a child with the disease. So now we need to figure out Michaela. And again, this is one of those where we know that this isn't her, but she could be either AA or AS. And so answering that question is going to require you to address both possibilities. So if she's homozygous for normal hemoglobin and Jerome is a carrier or has sickle cell trait, then there's a 50% chance that they would have a child with the trait, but a 0% chance that they're going to have a child with the disease. However, if she is also heterozygous, then there's a 25% chance that they could have a child with sickle cell disease. So sometimes when you're not sure, you should give both options. All right, now we come to the last part. Here's where we're going to do the chi-square statistics. So make sure that you've watched the other video I posted on how to use chi-square, and in particular how to use chi-square for genetics problems. Here we're gonna use it to see if the data that we're seeing in a population results in statistically significant differences between what we're seeing and what we would expect to see. So let's look. Multiple couples living in a small village in the eastern African lowlands, all of whom are heterozygous for the sickle cell allele, have 500 children among them. Of these children, 139 are homozygous, 279 are heterozygous, and 82 suffer from sickle cell disease. Are these data statistically significant? Meaning that do they diverge enough from what we would expect if it was just a normal dominant recessive inheritance pattern? So let's fill out this table. So my phenotype, genotype, so I could have normal, I could have trait, or I could have sickle cell disease. So 139 are observed to be normal. 279 are going to be observed to be heterozygous, and 82 are going to be homozygous recessive. Now, to determine my expected, make a quick Punnett square. So if all of the parents are heterozygous, I would expect 25% to be AA, 50% to be AS, and 25% to be SS, or have sickle cell disease. There were 500 children, so basically I'm going to multiply 500 times 0.25 for this, 0.5 for this, and 0.25 for this. And so I get 125, 250, and 125. Now to figure out my chi-square value, remember you're going to do observed minus expected squared divided by expected. And I am not doing this in my head. I actually already did this. So that is how I am getting these numbers. So if I just do observed minus expected, so 139 minus 125 gives me 14. And then if I square that, so 14 squared divided by the expected, which is 125, gives me 1.568, and I'm rounding. For 29, that gives me 3.364, and then 14.792. So to get my chi-square value, I'm just going to add all of those numbers up. And we can already see that this is kind of big, right? This number is pretty different. This one looks like it's a little bit different too. But let me add those all up, and I get 19.724 is my chi-squared value. And then remember the degrees of freedom, I'm going to take the number of choices minus 1. So in this case, I could either be homozygous for the normal hemoglobin, heterozygous, or homozygous for the sickle cell disease. And so I have 3 choices, so 3 minus 1 gives me 2 degrees of freedom. And then once I've got that, then I go to my handy dandy probability table. So this is on the last page of that worksheet. You also have it in your course packet. You can look them up online. All options. So I find my degrees of freedom. So I have 2 degrees of freedom. And then I'm going to look for my chi-square value. Remember that this is my critical point, my critical value. Just highlight that. So basically, anything that falls in this range would cause us to reject the null hypothesis. Which means that there's a significant difference between what we're seeing and what we would expect to see if it was just a normal Mendelian inheritance pattern. Anything over here, we failed to reject the null hypothesis, which means that there's not a significant difference, or there's not enough of a difference between what we're seeing in the population and what we would expect to see if it follows normal probability. Because you're dealing with just probabilities and you're dealing with human population, so there's lots of different things that could be going on there. So this kind of helps us to see, is it just random chance that I have individuals showing these traits, or is there a significant difference between what I'm seeing and what I would expect to see? So in this particular case, if I'm looking for 19.724 with 2 degrees of freedom, I am like way, way, way over here. There is definitely a significant difference between what I'm seeing and what I would expect to see. Because there's less than, this is the percent chance of getting that by random chance. So there's less than a 1% chance that this difference is due to just random chance. So my p-value is less than 0.01. And so I'm going to reject the null. which is basically that it's just normal dominant or recessive inheritance. There's nothing else going on, which means that something else is contributing to this inheritance. Now, that doesn't mean that, you know, A or S is dominant or recessive. It just means that something else, some other pressure is being exerted. And if you think about it, the children that have the greatest chance of survival are going to be those that are heterozygous, if malaria is present, or homozygous, if malaria is not present. And we see in the numbers that these two are higher than what we would expect to see, and individuals that have sickle cell trait are going to be a lot lower because they don't survive as long as their peers, because there's a lot of complications that come along with sickle cell trait. And so it makes sense that there's going to be a difference between what we would expect to see if there were no selective pressures and what we're actually seeing. All right, so last question, and this one we're going to look at two traits. So we're looking at blood type, and we're looking at the sickle cell trait. So here they're giving you, there's 50 couples with the same blood type and hemoglobin genotypes that live on a small isolated Pacific island with very few mosquitoes. So I shouldn't have a strong heterozygote advantage in this particular example. All the individuals are heterozygous for type A and have sickle cell trait. The 50 couples have 224 children, and these are their resultant genotypes or phenotypes. So the phenotype, I could have type A and normal, which would be AA, type O and normal, which would also be AA, type A and sickle cell trait, so that's AS, type A and type O and sickle cell trait, type A and sickle cell disease, and type O and sickle cell disease. This may look familiar because we actually did this cross earlier. So if you go back to that big table that we did, that I hopefully can find in all my notes. So many papers. Here we go. You can see that we've already figured out the probabilities for each of these. So I'm going to use those numbers to calculate my expected. So my observed is already given, so I've got 48, 18, 92, 33, 27, and 6. Now this is one way to solve it. Another way to do that, and I have it here, would be to figure out the probability for blood type and figure out the probability for the sickle cell, and then using that information determine the percent chances or the probabilities for each of these, right? So type A, normal, so there's a 75% chance of getting a type A and a 25% chance of getting both normal alleles for hemoglobin. So I just multiply 0.75 times 0.25 and I get 0.1875, which is exactly how I did it. Then I can take this number and multiply it by 224, because that's the number of children, to figure out my expected values. So I went through and I did all of that, and I got 42, 14, 84, 28, 42, and 14. You could also do that just using these numbers right here, right? Because 3/16 is going to be 0.1875, and then you just multiply that, right, by the number of children, that 224, because you want to know how many actual children, so you're comparing the same things. I kind of cheated and did the math earlier. Here again, I'm rounding. So once I have all of these values, I'm going to add them together, and I get 13.583. So degrees of freedom, here I have 1, 2, 3, 4, 5, 6 possible phenotypes. So that would be 6 minus 1, or 5. And now I'm going to go back to my table. So I've got 5 degrees of freedom, and I'm looking for 13.583, so I'm going to work my way along. Okay, so it's going to be somewhere, it's going to be somewhere between 0.025 and 0.01, so 2.5 and 1% probability that difference is happening by random chance, and so we're going to reject the null hypothesis. There is something different here. These numbers don't align with what we would see, what we would expect to see. So your p-value is between 0.025 and 0.01. So you're going to reject your null. And if you look at the data, you're still seeing this issue here, right? There's really no advantage to being that heterozygote in these conditions, but there's still a disadvantage in having sickle cell disease. And so it makes sense that you would have a divergence from the expected values if there was no selective pressure. So there's still, it makes sense, these guys right here are at a disadvantage, and so they're going to be lower, if there's going to be a lower incidence in the population. So I hope that was helpful. If you have any questions, be sure to email me, and I will get back to you. Bye.